Hey guys, this is Kendall Terry, and this is a look at genetics, specifically Mendelian genetics. Now, genetics deals with a term called heredity, which is the passing of traits from parent to offspring. And really, that's what this whole lecture series is about. So let's look at here a foundation of what we're talking about. So if we're talking about heredity and we're talking about traits going from parent to offspring, then we need to understand a little bit of background that was used up to this point. So the background here is that before Gregor Mendel, everything was believed to be a blending. So you take one parent and you mix them with the traits of the other parent and you get this offspring. Okay? So the offspring is just a blended version of the parents, which has some credibility, but there was some things that were lacking and missing, and there were some anomalies that needed to be described. So Gregor Mendel looked more at heredity and genetics, and because of what he did, he's called the father of genetics. So let's look at kind of what he did. So Gregor Mendel looked at a garden pea, and he did some experiments with the garden pea plant. Now, this garden pea plant is real easy to grow, and it gives nice, clear results and stuff, so it was a good choice for Gregor Mendel in this experiment. So he looked at several different traits, but for this example, we're going to look at flower color. So he took purple flowers, and he made sure that every time they reproduced, they only gave purple flowers. That way, they only expressed one trait of importance. Then he took another plant that produced white flowers. It was still the garden pea, but it just produced the white flower, and he reproduced it several times and made sure that it only produced a white flower. And then took these two plants and he cross-pollinated them, meaning he took the pollen from one and put it on the white flower, and he took the white flower pollen and he put it on the purple flower, and he looked at their results. So that first cross, we call that the pea generation. He took two what we call perennials or true breeders, because that's all they produced was purple or white, and he mixed them and crossed them and produced the first filial generation, or just simply the F1 generation. In the F1 generation, all of the plants were purple. Now that kind of goes along with a blending idea, but he didn't stop there. If he would have stopped there, then everybody would have said, yeah, it's just a two blending. You take purple and white, you put them together, and you really just get a variation of purple. But he took it a step further, and he allowed that F1 generation to what we call self-pollinate. Now, this happens in pea plants because their flower is closed, and they don't have to use pollinators. They don't actually pollinate with insects or birds or other things that typical flowers do. They keep their pods closed, and they just pollinate themselves. So they're able to self-pollinate, and he allowed them to self-fertilize or self-pollinate. And he produced a bunch of offspring that we call the second filial generation, or just simply the F2 generation. In the F2 generation, he got a bunch of purple flowers, which would have been expected, but he also started to get some white flowers. Now, one of the things Gregor Mendel did that was important to his experiment is that he added math to it. A lot of other people did similar experiments to Gregor Mendel, but they didn't take the math aspect to the degree that Gregor Mendel did. What Gregor Mendel noticed in his thousands of plants that he reproduced in these experiments is that when you put math to them and start putting them in ratio form, 3/4 of them, every plant he reproduced in that F2 generation, were purple. 1/4 was white. Now, these were numbers that continued to be repeated, so it gave credibility to this. This wasn't just a fluke chance that 3/4 were purple, 1/4 was white, but every time 3/4 of them are purple, 1/4 of them is white. And that is very interesting in his results, because it was a 3-to-1 ratio. This white trait would not show up if it was blending. So there had to be something else to go along with it. And that really gives us the foundation or the jumping off point to understand Mendelian genetics. So really, to help make all of this blossom, we need to understand some key terms. And the first key term that we're going to talk about is what we call alleles. I'm going to box this in right here so you know what I'm talking about. Alleles. Now, if we're talking about alleles, what we're talking about are different versions of the same trait. And this is what Gregor Mendel looked at. He looked at alleles. So he looked at the purple color and the white color. The purple is an allele for color of flower. The white is an allele for color of flower. So he looked at different versions of a particular trait. In this case, different colors. Now, he looked at other things too. Sometimes he would look at shape. Sometimes he would look at height. But there always had to be at least 2 different alleles for that trait, and then he would compare the results. So when he's doing this, he saw that some of those traits were expressed frequently. Let's try that again. He said some of these traits are expressed in the F1 every time. And that's the only trait that's expressed in the F1. So he referred to that trait as dominant. He said that allele has to be dominant over the other one because the purple parent is giving a purple allele, the white parent is giving a white allele, but all you see is purple. So apparently the purple will dominate the white or hide it from being expressed. So if purple is around, then purple is expressed. So that is a dominant trait. The other term he called recessive. He said the trait that's being masked in the F1 but reappears in the F2, in this case white, is a recessive trait. And the only way you see that is if the only allele present is the white allele. So let's look at actually how we write that. So what we use are letters, and we use capital letters for dominant, lowercase letters for recessive. So the P generation, or the parents, would have been big P, big P. That would have been the purple flower. And he crossed those with little p, little p. Now the reason why we know this is because the purple plants, every time he crossed those to get the parent generation, every time he allowed those purple plants to reproduce, they always produced purple flowers. And that's why he called those true breeders. Okay? The white flower, the only way white is expressed, remember that's recessive, so the only way white's expressed is if all they have is the white alleles. And he hypothesized that each of them, that they would have 2, one from mom, one from dad, which we know to be the case today. So here's your P generation, or your parents. Whenever you cross those, this parent here will give to their offspring a capital P. This parent here will give to their offspring a P. So now you end up with an offspring that is capital P, little p, for the alleles. Well, if the capital P is present, according to dominant and recessive, then it will be expressed. And this is exactly what we see. So the big P is present. All of these will have to be purple. And the F1 generation, all of them were purple. So that's correct. Then when we get down to the F2 generation, and we'll look at how all this works out, but in the F2 generation, there's 3 possible outcomes. There's a big P, sorry, this is kind of interesting in writing, the big P, little p, and the little p, little p. So there's 3 possible results. Now these two, big P, and big P, little p, are both going to be purple. The little p over here are going to be white. And so we say, according to this, yes, you will get white showing back up. And I'll show you exactly how those results come a little bit later. So this was his F2 generation. In the F2 generation, we end up with more purple than white, but white does reappear. And it supports that dominant and recessive thought. To help us understand and use some of these terms, we use some key terms that you can see over here as well. The first being heterozygous. Heterozygous are what we see in this F1 generation right here. A big P and a little p in one individual. Hetero means different. So they have different alleles for the same trait, the dominant allele and the recessive allele. So we say these guys here are heterozygous. Also, these guys here would be heterozygous. The homozygous one would be the ones that have the same. Homo means same. So big P, they have the same allele for that trait. So big P, that's homozygous dominant. Little p here, that's homozygous recessive. The last thing to understand is the difference between phenotype and genotype. Phenotype is when we talk about really what Gregor Mendel looked at. He looked at the color, purple and white, or you can think of it as a picture. That's its phenotype. Okay, so if I'm describing what it looks like, I'm describing to you its phenotype. If we go with genotype, now we're describing what you actually see over here on the screen. Big P. What are the actual alleles present? So this one has a big P allele and a big P allele, or a purple allele and a purple allele. That is its genotype. Here, they have a big P allele and a little p allele, or a purple allele and a white allele. That's its genotype. So there's a clear difference here. Make sure you understand that difference.